Okay. Yeah, I'm Dale Andersen. I'm at the Carl Sagan Center at the SETI Institute. And my research interests are, my background is as an aquatic ecologist and a limnologist, and obviously in exobiology. I've been working in and around the exobiology program since the mid-'80s, actually starting to work with Chris down in the Antarctic those years. But I'm interested in looking at life in extreme environments, and I've picked the polar regions as my area of interest. I've been working in Antarctic Lakes since about 1978. We opened up the first lakes in the dry valleys in terms of going into the lakes themselves and seeing what was growing on the bottom by diving into these lakes. At that time, it was really difficult to put a small camera down into these ecosystems because they just didn't exist. It was easier just to put us down. So we developed the techniques that allow us to explore these areas, and I've just been doing that ever since. So more recently, I've been working around the continent a bit. I've worked in the McMurdo Dry Valleys quite often. I've worked in the Bunger Hills, which is another ice-free area to one side below Australia. But more recently, I'm working in the mountains of Queen Maud Land, which is south of Africa. So we go down through Cape Town, South Africa, fly down on a Russian Ilyushin 76, snowmobile out into the mountains, set up a camp for a couple of months, and a very, very windy, robust environment. And there's a remarkable lake ecosystem there. And having been one of the few people that have been able to dive in all of these lakes in the Antarctic over the years, we're always looking for something new. And I have to admit, when we got to this particular lake, we weren't sure if it was going to be new. Maybe it's just going to be sort of the same old lakes that we've seen around the continent. And it'd be interesting and it'd be fun and exciting, but it wouldn't be really novel. And it turned out that it was just totally different. If we look at the lakes around the Antarctic, you get a continuum. So if you're on the very coastal sides, you'll get lakes that lose their ice covers during the height of the summers. If you go into some place like the McMurdo Dry Valleys, you get perennially ice-covered lakes, and the ice is anywhere from 4 to 6 meters thick, or in some cases you can get 19 or 20 meters thick. And those are pretty sealed lakes. It makes a big difference to have that big, thick ice cover on top. It reduces the amount of light that comes through the ice cover. It restricts the movement of gases in and out of the lake. So it has a profound effect on the biology and the ecology of these ecosystems. When we went to Lake Untersee, which is where I'm working now, we set up camp there, and the first thing that we noticed that there was no outward evidence of streams that came into this lake. And of course, a lake has to have some way of a mechanism of recharging it, or else it just evaporates away. In the dry valleys, we always have, at the height of the summer, the glaciers perk up and start to melt, and you get lots of runoff coming into the lakes, so that's pretty obvious. Here, there was just no examples of water coming in anywhere. There was no channels coming off the sides of the glaciers. So it was clear that this was being fed through glacial melt and subglacial activity, which is pretty unusual. And as it turns out, this lake didn't have any moat formation around the edges either, so it's really, really quite sealed. So in terms of a surface lake, it's probably the most sealed surface lake on the Antarctic continent that we know of. After that, you would tend to go underneath the continental ice and start having lakes like at Lake Willems or Lake Vostok, which is under kilometers of ice. So this turned out to be pretty exciting for us. As a matter of fact, I took Chris with me on that first expedition, and the first thing that we did was go out and drill through the ice to figure out where we're going to put a dive hole and to get some drinking water. And as we did that, it started geysering water out of the lake. We had a plume of water coming up. There was gas coming out. It was just remarkable. So from that point on, I knew this was going to be a really cool place. Eventually, we made the dives. We discovered that the microbial communities in this lake were quite different. All of these lakes tend to have cyanobacterial mats that grow on the bottom and colonize the bottoms. And this particular lake has morphologies that are quite different from any of the other lakes that we've looked at. We have large conical stromatolites that are forming on the bottom. These are large cones that come up off the bottom. As far as we know, it's the only place in the world that we know of for sure where this particular morphology of cyanobacteria has been expressed. And we've kind of thrown that out for somebody to show us that we're wrong. The other part of the lake on the other side, though, it's a very deep lake. It's about 170 meters deep right next to the glacier. It comes up along a sill, drops back down to 100 meters deep on the other side. In that shallower 100-meter basin, the water column is quite different. It goes anaerobic when you get down below about 80 meters. And at the bottom of the lake down there, you get methane concentrations that hit 21 millimolar, which is about the highest level of methane that you'll get in a natural ecosystem unless you're right above a marine clathrate out in the ocean. So we've got a really nice natural laboratory here to help us understand paleo lakes on Mars and also a really nice laboratory to understand energetics and microbial ecosystems that occur below thick ice covers in a sealed environment. And then just as luck would have it, right next door to this lake is an old paleo lake basin. As the glacier that is damming this lake pulled back, it actually pulled back far enough that there used to be another lake off to the east a little bit, and when the ice pulled back, it cut off the water supply, and that lake just dried down over a few hundred years. And so now we've got a microbial ecosystem that's been left and buried in the sediments that are remaining there in this permafrost area. So it's a great laboratory for us to understand how biosignatures are preserved in these kind of environments. And I'll just stop there.
Okay, Steve, introduce yourself and be slightly shorter.
Yeah, my name Fight. Excellent. Yeah, my name's Stephen Benner. I'm from North Central Florida at the Foundation for Applied Molecular Evolution and the Westheimer Institute for Science and Technology. I'm a chemist by training, but for the purpose of this session, I'm interested in natural history on Earth. We can go take modern biology, and by analyzing it, we can make models for what ancestors look like. And of course, it's easier to make ancestors of everybody in this room than it is to make ancestors of you and the plants that are outdoors. But at the molecular level, one can do alignments of protein sequences and infer the sequences of ancient proteins and bring those back to life. And then from those, draw inferences, not about Jurassic Park quite, but about what life looked like, what life did to earn its living, and what life environments were, say, 2 billion years or 4 billion years ago, as far back as we can go. Now, for the purpose of searching for life, one of the great things you can do is say that, well, we know actually from this kind of analysis that the biosignatures of today were not the same as the biosignatures, say, of 2 billion years ago, and not likely to be the same biosignatures as 4 billion years ago. And so one of the games you can play whenever you have an idea for how to detect life in the cosmos is just ask whether it would have detected life on Earth 2 billion years ago or 4 billion years ago. So as an example, those people who study protein translation have an order in which various amino acids appeared in encoded proteins, and tryptophan is one of the last ones. Well, tryptophan is a bit unfortunate because it's actually the most fluorescent, probably the easiest to detect remotely or in a sample of an alien planet. So since tryptophan emerged late compared to other amino acids, if you set out on a mission to detect tryptophan, say on Europa or Enceladus, the problem is that, well, he wouldn't have been successful in detecting life on Earth 2 billion or 3 billion years ago before tryptophan actually emerged in encoded proteins. The same is also true, say, for lipids, right? You're very familiar with the lipids that you have, which are made by hooking together 2 carbons at a time. So you have a lot of the C10 lipid, a lot of the C12 lipid, a lot of the C14 lipid, a lot of the C16 lipid, but no lipids that have odd numbers of carbons in them. But of course, that's a very recent innovation in terms of geological time. It only emerged after translation, emerged out of the RNA world. It does not predate that. So if you looked for that as a biosignature, you would have actually had a hard time finding that evidence for life on Earth 2 or 3 billion years ago using that signature. And since we are expecting the life on Europa or Mars or Enceladus to be more primitive than us, with primitive defined in the human sense of the term, that kind of consideration is very important for us. So that's a place that I will start. So I end up being very much of a negative person whenever anybody comes up with a biosignature, and when it's based on what Earth has today, I always criticize it because that's not necessarily what Earth had at the time of its age or stage of primitiveness that we will be encountering when we go seek life on other solar system bodies.
I'm Natalie Cabral. I'm the chief scientist at the SETI Institute. I'm also an astrobiologist. My background is both in planetary science and earth sciences. What drives my interest is really exploration in a larger sense. When I arrived in '94 at NASA Ames, I knew that although I was interested in lakes on Mars, just for the same reason as Dale is interested in them, because they are good places for life, you could provide NASA or any other agencies with the greatest sites in the world, but if it's not safe and you cannot have a mission, you crash a mission, you don't have a mission. So I knew that I needed to get my hands on understanding how engineers think about mission, about exploration. And this lady over there taught me a lot about that when I arrived at Ames, and got me started with rover and the Mars Lookout Field experiment. So I learned from Cabrol a lot, and then move on to a thought process that what am I looking for, and how am I going to be looking for it, and what kind of resolution, what kind of instruments do I need to do that? And basically you go through that process. These were the questions that Penny was talking about this morning. In doing so, I was interested in lakes on Mars. We were pushing very much at the time in the 90s for crater lakes. Interestingly enough, everybody was talking about water on Mars and channels on Mars, but Dale might remember how tough it was to actually convince people that water was ponding somewhere. And so we did that, and then we had to look for traces of that on Mars. I was looking at the time at Viking images. They were not the greatest, but we started with impact crater lakes. And then the next question was, well, if there were lakes on Mars, what was there? And this is where the terrestrial analog starts. And this is where you're looking for sites that are looking like what you think Mars was looking like in the past. So to do that, you have to go to places where you have a little bit less of an atmosphere that you have on Earth, that you have high UV radiation, where you have volcanic environment, hydrothermal system, etc. And this is how I ended up in the Andes doing a lot of field work, and I'm still doing a lot of field work. I'm right now a PI on an NAI team under Penny. So this is really what drives me, trying to understand, going through those thought experiments. If I was a microbe on Mars, 4 billion years ago, where would I like to be? And this becomes also an interesting question, because as we go, so I got involved, I was lucky enough to get involved with the Mars Exploration Rover mission, and I got to see Gusev and Meridiani from the ground. So these are sort of life-changing experiences, but also that taught me something very important, and also the work that I've been doing in the field in terrestrial extreme environment in the Andes are telling me something, is that we have, for the past decades, looked at how habitable the solar system is. And I think that we are all pretty convinced that the notion of habitable zone is so yesterday, all right? You have habitable environment, but those habitable environment can be so disconnected in space and time from the habitable zone. And I think there is a reaction right now from the scientific community who wants to just break the confine of these boxes we have been putting ourselves into. It's not that we put ourselves in boxes. We needed those, as you said, Penny, this morning. We need those to get started, but we should not be prisoners of these notions. And so today we are going to start to look for life in the solar system, because we're convinced that it could be there. And we have to move on from the notion of habitability to something else. Once you put life into the equation, it's not habitability anymore, it's coevolution. And the biosignature we are going to look for, these are signature of interaction between life and their environment, and they change the whole game. And every single planet, every single moon you are going to visit is a very unique planetary experiment. So I don't know if, as Alfonso was putting this morning, the whole prebiotic chemistry is the same everywhere in the solar system. No matter what happens, he will end up doing maybe something else because it happened on Mars or on Titan, etc., or it won't happen. But I think that we are at a critical point in time where we have first to just get rid of those boxes and be a lot bolder about our hypotheses, because they have major implications on the way we think about exploration. You are not going to be able to think about exploring Mars as if you were exploring Earth. Mars was very, very early on a very extreme environment. MAVEN has changed everything. The data that we've received from MAVEN that says that 4.1 billion years ago the magnetosphere was gone, and the atmosphere was already going down 3.9 billion years ago, puts Mars in the extreme condition at the surface very, very early on, early Noachian. And when you look at the distribution of life in extreme environment on Earth, which is probably even better than anything that Mars known very early on, it's stochastic. And so we have to think whether what we are thinking in terms of exploration, big rovers and driving and going to the places we want to go is the best approach. I'm not saying it's not, but we'll have to rethink that, I think, in that light. Passing on.
Cynthia?
I'm Cynthia Phillips. I'm a planetary geologist at the National Jet Propulsion Lab down in Pasadena, California. And my favorite place in the solar system is Europa. I'll come out and admit it, even though you're not supposed to tell any of your children they're your favorite. Europa's my favorite. None of my children are named Europa, so it's okay. But I'd like to revisit a bit of what we talked about this morning. So we heard an excellent case for why Enceladus is so fascinating. We heard about why Europa was fascinating. We heard about Titan. And one comment was made that it would be really amazing to find life beyond the Earth, to find life existing somewhere else in our solar system. But I think that there are two very different cases that you can make. One is if you find a place where life currently exists that's not the Earth, but you can trace that life back to the Earth. I don't know, I think I'm with Chris. I think I would be really disappointed if that's what we find. If we go, if we find life on Mars, if we find life on Venus in the clouds, and we trace it back and it has, the same DNA as us here, we can prove that it wasn't just contamination on our instrument, planetary protection did its job. It actually is, terrestrial life that came here or Venusian life that, came and seeded Earth. I would find that disappointing. I mean, I'd still be excited, but what I really want is that second origin of life. And just because I think that that's really where you're in sort of the earth-shattering, paradigm-shifting, life becomes abundant in the universe. You go from having life start once in our little dinky podunk solar system and the unfashionable arm of a spiral galaxy, right? Okay, life could start here once, but if we can show that life started here twice in our own solar system, and then you look up at all those worlds in the sky that Kepler's been finding, all those exoplanets, if life can start twice here, then it's gotta be everywhere. And so that's why when I think about studying the outer solar system, and in particular Europa, which, okay, we've already admitted is my favorite place, Europa presents to me the best possible case for finding life beyond the Earth that didn't come from the Earth. We know that life can be transported, it can be swapped around in the inner solar system, but if we find life somewhere like Europa, and I think that Europa really has the best chance for, it has conditions for life. We think that there's this global ocean, like Brittany talked about, and it's been there, it's been stable throughout geologic time. So if there's an ocean on Europa, it's likely been there for 4 billion years, that it's a stable, clement environment. You look at the radiation on Europa's surface, which, it makes exploration of Europa more difficult, but that's an energy source that provides fuel, oxidants, organics for a potential biosphere down in that water layer. And so when we start thinking about future missions like Europa Clipper, which is going to investigate the habitability of Europa, that's going to be a huge step forward in terms of understanding how thick is that ice layer? What is the actual composition of the non-ice materials at the surface? I once gave a talk when I was at the SETI Institute. I don't know if Frank is still here, Frank Drake. I gave a talk where I referred to the non-ice materials as contaminants. And Frank very nicely corrected me afterwards and said, no, that's the interesting stuff. Don't call it contaminants, and he was absolutely right. So, the non-ice components of the surface. So we'll learn what those are actually made of with the Europa Clipper mission. And then there's this great planned, this mission concept called Europa Lander. And this is a mission that Brittany alluded to a bit, but so you actually land on the surface of Europa and you bring a full-fledged mission. So you're doing in situ astrobiology. You're actually scooping up the surface, you're digging below the radiation process deposit right at the surface. The plan for the mission is to go down about 10 centimeters but below the bulk of the radiation processing. You scoop up material, you process it on the board of the spacecraft with a model payload that includes things like a GCMS, a RAMA, and a microscope. But then the spacecraft also has full-featured instruments to characterize the environment, to characterize the geologic context for those measurements. So you bring along a geophone or a seismometer, and you bring along a great camera. And that gives you that in-surface, that surface, that in-situ perspective on what does Europa's surface really look like? What's actually going on there? How do these processes, like Morgan mentioned, it gives you that human scale perspective on all that geology that so far we've only seen from orbit. And I would posit that we've never seen a world that has a surface like Europa before. All of the places that we've landed are fundamentally Earth-like, except for the moon. So we've landed on Mars, we've landed on Venus, we've landed on Titan, but those are three worlds that are shaped by aeolian processes and they're shaped by fluvial processes. And okay, the actual materials and the temperature ranges are very different, but what's going on the surface, it's fundamentally a triumph of planetary geology in that these same processes go on, they produce the same kinds of surface features, but they're fundamentally similar. And so we landed on the moon. The moon is fundamentally non-Earth-like. It's shaped by completely different processes. It's a surface that's exposed to a vacuum. But our images of the moon, our mental images, are all shaped by those astronauts walking on the moon. So we can relate. When we look at pictures of the moon from orbit, we have this mental image of an astronaut standing next to a big boulder, or of a spacecraft landed on the surface, of what that footpad did when it hit the surface. We have no idea what Europa is going to look like from that perspective. And so that right there, I think, is the revolution, not just in biology, but also in geology, that we'll get from landing on an icy world like Europa. So that's why it's my favorite.
Me that Europa Lander is a good idea.
and for anyone who wants to hear more about Europa Lander, Morgan and Kevin Hand and I have some copies of the Science Definition Team report handy. If anyone wants a nice, big, thick bunch of reading for your plane ride home, please see one of us.
All right. Carol, last but not least.
Okay, I'm Carol Stoker. I'm a research scientist at NASA Ames Research Center, along with Chris and now Penny. So I've been struck throughout my career with why aren't we finding life on Mars? And it turns out that I started graduate school the year Viking landed on Mars and looked very deeply into what the Viking experiments were about and how they were interpreted. And sort of set me off on a course where throughout my career I've kind of had this theme of two things: human exploration of Mars and search for life on Mars. And I've been very struck at this conference so far that we've had these presentations about these target bodies that did not include Mars as places to look for life. And it brings to mind this contest that was held, or a prize that was offered by a French millionaire in the 19th century that was a prize for finding life on any planet other than Mars. And it was 100,000 francs, which at the time was a lot of money. And the reason Mars was excluded was because it was thought it would be too easy. And so I think that that's kind of where we're coming from here in this conference, that it's going to be too easy to find life on Mars. And therefore, we'll look at all these other places. So I think that because we did this mission to Mars to look for life on the basis of kind of a fantasy of what Mars was like that was based on the knowledge of Mars in the 50s and 60s, and pictures of Chesley Bonestell and Wernher von Braun, generating these sort of interesting landscapes that were complete fantasy. We went directly to a life search mission without really understanding what Mars was like at all. And so now that we've been doing flight missions that have kind of assumed there's no life on Mars and had no instrumentation to detect it, we're getting better information about what is Mars really like. And we're starting to realize that there are environments on Mars, modern environments, that are habitable in modern times. Maybe not right this minute, but plausibly habitable environments. And one of those environments is the ground ice. We now understand that there's ground ice over a lot of the surface of Mars, down to 40 degrees latitude in the northern hemisphere. Most of the northern hemisphere is ice saturated and at basically right near the surface. So the instrument that can detect this is a neutron spectrometer that's been in orbit actually for like 2 decades now. And that neutron spectrometer is only sensitive down to the top meter. So the fact that we're seeing ground ice in the top meter down virtually at all longitudes down to 40 degrees south or 40 degrees north latitude means that there's basically a frozen ocean sitting right there. And that we also know that Mars goes through these huge obliquity variations so that the insulation that's falling on the northern hemisphere varies over time. And we happen to be in a very bad time right now. So we are in essentially an ice age in the Mars northern plains. But as obliquity increases, and it increases in the last 10 million years up to 45 degrees obliquity, that ice can melt. A lot of that ice goes through thermal cycling down on very short time scales compared to how long life could be preserved if it's sort of in frozen condition and not metabolizing. So I've been advocating for a number of years since the Phoenix mission, which I was involved in, that we should go back to the Phoenix landing site and we should drill and we should sample that ice to detect life. For an exploration that has a fairly good probability of getting a yes/no answer. So I'll stop there.
Okay.
Yeah, so we're going to try to kick this off by irritating people with a question. And then I think since we're very close to our introductory time being up, that we'll open it up to questions and dialogue with the audience, because I want to see, these interactions. So the annoying question that occurs to me is that we have a conundrum. It's really a comment, but I would like your responses to that. We have a conundrum in that we have one example, not only of life that we know about in our solar system, but the reason we know about it, besides the fact that we live here, is the fact that it's really conspicuous. It's really a planet-dominating process. If we have life, either a residual biosphere on Mars, or we have these sealed roofed systems in icy moons, this means this is a very different kind of animal. I've been calling that a type 2 biosphere, that it is cryptic, that it is not obvious on the surface. So the conundrum is you can only easily detect a system that is a planet-dominating force. And if it is cryptic, then it becomes a very difficult proposition to actually find it. If you were looking for another analog of our solar system out there amongst the exoplanet suites that we have, what do you think would be the way that we could identify solar systems that may have no type 1 obvious Earth type signatures? Is there any way to actually identify a cryptic subsurface Mars residual biosphere? Is there a way to identify life signals from an icy moon?
Cynthia?
Cynthia's nodding.
So that's
Right.
a tough question.
Yeah.
And so when we look at the Earth, I know that Carl Sagan did a famous search for life on Earth with the Galileo spacecraft. And so that's kind of our best analog for how we might explore Earth as an alien world and look for signs of life. And I believe that, I haven't read that paper in ages, but it's a lot easier to find evidence of a technological civilization than it is to find evidence of microorganisms. At the SETI Institute, we had there's the search for extraterrestrial intelligence, and then there's the Center for the Study of Life in the Universe. And so I used to call that, there's the intelligent life people, and then there's the stupid life people, right? So, and I was always a stupid lifer, right? So if you're looking for stupid life, you're looking for life that doesn't send out intentional or even unintentional signals into space, you're basically stuck looking for things like, for example, interesting, is there chlorophyll in the clouds, right? Or is there an imbalance? Are there, like methane on Mars, for example? Are there materials that you can detect through remote sensing that are evidence of some kind of cycle or some kind of imbalance or something of that sort. I think also when you're looking at exoplanets, it's very hard to find something like Mars. Let's say that Mars had an old ancient biosphere, and now maybe there's remnants in a few sort of clement places, or same thing with Venus, right? Where there's kind of, there was a once abundant surface life, and now it's kind of hiding in a few little niches where it can still survive. That's going to be really hard to define when we're looking at exoplanets, unless we can, invent a warp drive and go there, which I guess we're doing tomorrow. So we can postpone that discussion.
Right, and I'm waiting
But
for the
when
answer.
you look at, when you're looking for icy moons, there I think if you find a system that has giant planets, I think that can be a good indicator that there could be icy moons there, because we have 4 giant planets in the solar system. All 4 of those have large moons, 1 or more around them, and we know in our solar system it's resonances among those moons. So I think it's not crazy to think that maybe we could look for exomoons. And that might be a way of saying, okay, maybe there's a good chance that there could be one of these kind of more cryptic biospheres, a type 2 biosphere there.
So if we, oh, sorry, Chris, I had a follow-up, and that was, suppose you imagine 50 or 60 years from now, this is an optimistic interpretation, that bingo on life in Europa, bingo Enceladus, maybe even bingo Mars. Does that mean that it's
Venus.
a slam dunk? Well, Venus, okay. Venus.
Titan.
Titan.
I mean, is it a slam dunk that there's
every suite of planets out there? Yeah.
I would say yes. I would say boxes checked. Okay, if we find multiple biospheres within our own solar system, again, you look up at practically every star in the sky. We know that, over the course of my professional career, exoplanets have gone from being something that crazy people looked for to something that everyone takes for granted, right? I mean, it's not that long.
Yeah.
You look at all those stars up there, you know that probably the vast majority of those stars have planets. And if we have multiple sources of life on multiple places in our own solar system, yeah, it's going to be everywhere. It's going to be teeming.
I just would like to add
Go ahead,
maybe something to this. It's part of the exploration process, and we are all so excited. Our mind as scientists is already out there, 200, 1,000 years, ahead of the time.
All our minds are
But,
out there.
we have to do the homework, and we have an entire solar system to do that. Right now, we're at this incredible time where we are putting, all of these spacecraft out, but we are just taking baby steps. And we're starting to project ourselves into an exoplanetary system out there. What I would say is that we need to learn, how we function. We have a fantastic natural lab right here. It's our playground. We need to learn what a potential biosphere can be in any single place of the solar system. You can develop a model of a biosphere on Europa or on Enceladus, Titan, Venus, whatsoever. We need to understand what makes a biosphere work. And we have the data. We are starting to have the data. Now we will have more. But we also have fantastic tools, data analytical tools with machine learning that can allow us to project models. And once we are feeding those models with the data we have, then we can start extrapolating, because right now what we are doing is speculating, and this is very different.
Well, that's a good key to go to the audience.
Go to the audience.
If you have a question, raise your hand. Let's start in the beginning so we can practice the throwing on small distances first.
Make sure you hide your coffee, move your laptops.
Carolyn, you ready? Good catch.
Is this it? Yep. Look at that. And I feel like an idiot.
Yeah,
You are.
I hate those
You have
things
me
too.
talking into a box.
Talk to the box.
Filming this.
Talk to the box.
Hey, I just work here.
Get out of the box. Okay. I'm not going to do this. Can everybody?
Set it down on the shelf. Set it down. Just set it down and talk.
Put it down.
Yeah.
Okay, now talk.
Broadcast.
I thought what Steve had to say was absolutely fascinating. I guess I heard you say this once before, and I may have been too busy and didn't catch it the way I just caught it now. You're saying that all this, or maybe you're saying that all this talk about biosignatures, what we ought to go look for, and there's endless talk about all this, and people debating what are the best ones. Could everybody be on the wrong track because we're talking about biosignatures today, the way the biosphere is today, And have you been trying to tell people, no, 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 we should be looking for this and not that?
Unsuccessfully.
Unsuccessful.
This paper did get published. It's in astrobiology.
Well, okay, I mean, let me make a comment.
This is very
That's why he's on the panel.
One of the,
good.
We didn't just randomly pick his name out of a hat.
That Steve guy, let's
We need somebody with a B in their first initial. A, B.
One of the questions you have to ask, this is also relevant to what Penny says, is whether you believe that Jeff Goldblum life finds a way, right? So the question is, what? Yeah, Jurassic Park. Sorry,
Triassic. No,
these are
not
American
Triassic.
allusions that may not translate to international cultures. But the question is, on Earth, in fact, this is what Chris McKay's work, a lot of it shows, is that wherever you have thermodynamic disequilibrium and water, you have life. Life has found a way to occupy that zone. This is what Penny has shown in some of the weirder places you saw a picture of her earlier with gloves on. And so one of the questions you have to ask is, if the life is not dominating the entire planet, why is it not dominating the entire planet? Since there's thermodynamic disequilibrium, I guess, everywhere from the solar radiation. Now, one possibility is that the disequilibrium has been run down, right? So Mars, because it's not a dynamic planet, you could make the argument that we've lost the equilibrium. And so that's why you don't have life dominating the planet, because maybe it did, and of course life itself will run down the equilibrium, the disequilibrium, if it gets a chance. The other possibility is that, of course, you do need a fluid, a liquid, a water on Earth, and maybe you just don't have any water on Mars. That's the other argument. But back to your point, is absolutely yes. I mean, Earth has gone through a number of episodes. Now, you don't have to believe all of them, but there are some people who believe that translation, that is making proteins from messenger RNA and transfer RNA in a ribosome, arose later than Darwinism. That is, the first Darwinian systems on Earth did not have encoded proteins. Now, the facts are that if you have encoded proteins, you really have to have homochirality among all the amino acids, and that's what we see today on this planet among the protein used in proteins amino acids. But in point of fact, if the Earth 2.5, 3 billion years ago used RNA as its only genetically encoded component of biological catalysis, there is no reason for the amino acids to be homochiral. In fact, you have proteins today on Earth like grammacidin, which are not made by translation, where half of the amino acids are left-handed and half of the amino acids are right-handed. So exactly what we're saying is that if you assume that you're going to look for homochirality among amino acids, what you're assuming is that there is an encoded protein. Now, that's fine. It may very well be, and it's certainly true today on Earth. But if you go back 3 billion years to the RNA world, if you believe in the RNA world, then you conclude that there wouldn't necessarily have been homochirality among the amino acids, and therefore looking for homochirality in the amino acids would not have detected life here on Earth 3 billion years ago.
So what should we do?
Look for it anyway. That's my answer. Carolyn, can you pass the blue box over there, and then we'll just work down and back without throwing it too far.
I wanted to draw some lessons from studies of the physical universe, where we knew of some laws of physics, nuclear physics, for example, here on Earth. And then we looked out at the universe, that's exploration, and we did find new substances like dark matter and so forth. But it was very useful to have actually laboratory experiments to enlarge our imagination. And this is a component that was not brought up. My question to the panel is, why isn't that one of the main facets of astrobiology, to have laboratory experiments such that we can, for example, test other fluids other than water, test extreme conditions, test completely different chemical networks? And I think it should be a major path of exploration without going to the stars, without going to other planets.
Yes, let me just interrupt, though, that yes, yes, and yes, and that's the next session. Session 2.
Well, no, as far as I know, there aren't many laboratory experiments trying to...
Oh, well, ask that question. There should be.
It's very different than the one we see naturally occurring on Earth.
Yeah, but the people like Lynn Rothschild and the synthetic biology mafia are working on this. That's what they're trying to do. You are right.
Is it a third of the community?
Maybe. I'm trying to keep it down.
So why doesn't it get
There's only so much funding.
Just like in physics. In physics, it's a major component of the funding.
Right.
I mean, it goes to laboratories that explore physics without going to the universe to find whether it's satisfied there.
Maybe the synthetic biology mafia will take over all the funding, like the experimental physics folks do, but so far they haven't succeeded. But you're right that there's got to be a balance, but that is a topic that should be discussed in the next session. And in fact, I'm on the panel in that session, so I can promise you we will address that.
Okay, so let me rephrase it. Different liquid or different form of life, how do we enlarge our imagination? Or do you think there might be a different, other than water, let's say ammonia or something else, in which...
I'm part of the synthetic biology mafia here, so I would hire you as my marketing agent tomorrow. You should come by for a contract.
But in fact, Steve is being modest because he has been one of the main proponents of thinking through exactly what you're saying. And in fact, his talk tomorrow is supposed to be on that topic. You never know with Steve if he'll actually talk about what you ask him to, but that's what he was asked to talk about tomorrow. So maybe we could go to the next question.
A question really for the enthusiasts about Europa and Enceladus. If you found very simple life, would you be much more surprised to find more complicated life, like in the ocean vents of the ocean, because it's been 4 billion years of evolution. So if you have simple life, then is there any obvious reason why it should not become more interesting life?
I would love to find giant squid on Europa, and in fact, that's my secret goal in exploring Europa, is looking for the giant squid breathing holes.
Simple life?
Yeah. So unfortunately, while complex life is, of course, a possibility on ocean worlds like Europa, it's much more likely that there just isn't, the energy gradients just don't sustain it. There's just not enough fuel to sustain big, large, complex organisms. So we probably are just talking about little microbes, which I won't say are boring, but I definitely agree are not as photogenic as something like giant squid or something of that sort.
But let's take that question further, and maybe Natalie or Carol could talk about on Mars, are we expecting to find microbial life or big things? And then Dale, what do you find in these lakes? Do you find giant squid? And why or why not? So maybe Mars first.
Yeah.
Giant squid on Mars?
Giant squids on Mars frozen in the Northern Ocean.
There you go. You heard it here first, folks.
Yeah. Well, when you look at what I think, what is similar, although this is not exactly the same thing, is that I definitely believe that there was always a better chance for life on Mars in the subsurface. No matter when, no matter what, etc. So if you go deep enough on Mars, you have liquid water and you have energy, and you can have very stable environment for a very long time. So it might be difficult to develop a giant squid or anything that's large just because of the energy balance, etc. But I think that you can sustain ecosystems that are going to develop a lot of biomass. But I would still think that we are looking at simple microorganisms as well.
Did you want to
and especially since on Earth, it took like 3-something billion years to get to organisms that are much larger anyways.
But we do have giant microbial mats, right? Yes. So it doesn't have to be big individual creatures.
The colonies might be big by themselves, the habitats.
Yeah. Carol,
do you want to add anything on the Mars front?
No, I would agree with what Natalie is saying, that we didn't have multicellularity on Earth until we actually got an oxygen atmosphere, or we got enough oxygen in the atmosphere. So it's energy driven. So you need to have a very high energy source. The interesting thing, and again, the new data from Mars, it's a decade old now, is the presence of perchlorate. And it turns out the perchlorate metabolism is very similar to oxygenic metabolism. In fact, many facultative anaerobes can also use perchlorate, and it has a very high energy yield. But I think that it's very likely that perchlorate is concentrated on the surface. It probably isn't getting down into a subsurface aquifer, at least at concentrations like we see it on the surface.
Dale, Antarctic Lakes?
Yeah, well, if one assumes that Mars has always been quite cold, and even at its warmest time in history, it was more like the Antarctic, where we do have mean annual temperatures well below freezing, but we still have liquid water on the surface. Those liquid environments are covered by ice, which provides a nice protective cover for them. But these are all microbial ecosystems. and they're photosynthetic in this case. So we do have large amounts of biomass that form on the bottom. Now, originally, when we first started looking at these lakes, a lot of people had looked in the water columns and not really found that much. They were interesting, and depending on the lake you were at, they had different metabolic strategies. But the bulk of the biomass was not observed until people actually started looking down on the bottoms. And here we have these large colonial collections of microorganisms, building lots of different morphologies and structures that can get preserved in the environment over time. So that, that's something that we could look at if we're really lucky in some of these paleo lakes on Mars.
But Dale, let me push on that a little bit, because the explanations for searching for single-celled life that have been offered are evolutionary. It didn't start on Earth for a long time. But in these Antarctic lakes, that's not the explanation, because obviously there's multicellular organisms all around. So is there a simple way to explain why, say, Lake Untersee has only microorganisms and it doesn't have, say, algae-eating fish that would just love to eat all that algae on the bottom and would be very happy to do so? Is there a simple...
Yeah, right here on Earth, the obvious explanation is that there's an exclusion mechanism that keeps everybody else out. And of course, in the polar regions, it's the fact that they're very remote, it's very cold, and in these environments like the lakes even, if you compare the oceans with the lakes, for example, the oceans are teeming with life, but they've got a refugia that connects back into the temperate systems very easily, whereas these lakes are sequestered in places where life has a hard time getting to it, and it has a very difficult time surviving underneath those situations. So that ice barrier for the lakes provides a very, very nice exclusion mechanism for them.
So if you were to seed multicellular organism there, it might grow. And I realize that would be illegal.
Well, no, actually, you can actually work your way back away from the dry valleys or from Untersee, go towards coastal regions, and the trophic levels go up. So all of a sudden, you're starting to get multicellular life. You'll start getting mosses, for example. You'll start getting zooplankton. And that's about as far as it goes. You don't typically see any fish, for example, in any of these lakes around the coast.
Okay, Jim, did you have a question? And then over there.
From an astrobiology definition of life, it has three attributes. That's how we identify it. And one, of course, is it metabolizes. That's what requires the liquid. That's why following the water and looking at, what methane might contribute to weird life is a great approach. And then it divides, reproduces, and evolves. So the evolution part is what's really exciting, I think, because if in panspermia we've got life like us starting in Mars, it will have evolved very differently. And would we be able to recognize it in the sense of how that environment has changed so much over time?
Natalie?
Well, you have the three criteria. So physiologically, if you macrophysiology, right, there's a lot of weird-looking creatures on Earth, and we have come without much difficulty to recognize them as being alive. Now, it did take many, many years, including DNA sequencing at some point, to confirm, say, the tree of life, but certainly paleontology, well before there was any need for molecular analysis, was able to get the tree of life of macroorganisms more or less correct. So now you have to go back to microorganisms and say where they really have had to rely on molecular signatures to be certain we know the relationship between various types, because the microbe will evolve to be suited for the food that it's in its particular environment, even if it comes from a distant branch of the evolutionary tree. So we would, of course, if you were to be able to say, of course, then you have the other question as to whether this life on Mars is cryptic or not. But let's take your question. It's not cryptic. We see it, we have it in hand, and we can do a complete molecular analysis of it. Unfortunately, the first thing we have to do is kill it, which is why all the aliens on Earth are hiding right now. They don't want to be discovered. But presumably, we would be able to find sufficient numbers of chemical signatures that are common to the common ancestry of the life that went there. And that's because certainly when we look at the molecular structures of life here on Earth in what we consider to be mildly exotic environments, nowhere near as exotic as what, of course, you have on Mars, even when you lose the magnetosphere, we expect to be able to see commonalities that would include essential components like the nucleotides, or even more general things like cofactors, metabolism, metabolic pathways, and of course chirality.
I think that actually could be a very interesting pathway for planetary protection, taking very simple microorganisms here and run them through lab experiments that are taking into account what we call poly-extreme interactions. We tend to apply high UV to some organism or desiccate them or do a number of things, one by one, but in fact they are living in a system where all of these elements, all of these extremes are acting at the same time. And so if we can take very simple organisms and run them through those poly-extremes and see how fast they are adapting or dying, etc., maybe get a number of signatures that we could recognize as potentially something that is Martian or something that is terrestrial. This may be actually an interesting pathway for allowing planetary protection to be a little bit more flexible by giving us some library of signature that we could count on and read as we go.
Do you want to comment on that?
Well, I just wanted to make a kind of an observation. I mean, when we think of extreme environments, as people, we tend to think, wow, these are really horrible places to live. And the microorganisms don't think that. They think it's great. Yeah, I mean, when we go to a place to look for life, I think if life has colonized that place, it's going to be really have taken over the place, and it'll be obvious. Life will exploit it to the limit, and it'll be there. Just like a good example of this, I think, was, and I'll bring up Emery Friedman again. In the 70s, Emery had made the prediction that he would find endoliths colonizing the sandstones in the Antarctic based on some of the work that he had done in the Negev Desert. And for years, geologists had walked over these rocks and never noticed anything. I mean, there was an exfoliation pattern sitting right on the surface of the rock that was developed by the microorganisms themselves. They never saw that. And you would think that that is actually one of the most extreme environments on the planet. Yet, if you open up the bottom of these rocks, they're everywhere inside the rocks. They're not just in one little spot. You don't have to look, forever for that one special little rock to find the life in it. You can go and open up almost any sandstone in this area and break it open and see that it's there, unless it's in an area that has experienced a mass extinction event in that particular zone because the water levels went down. But I think once you find life, it's going to be everywhere.
Hidden and yet everywhere at the same time.
Right. It's either going to be so hidden that we don't understand it at all, or it's going to be jumping up at us.
Okay, next question here, and then we'll go over to Rocco, and then maybe Ralph will try to get you in, and then we'll try to move farther back into the audience.
Planetary Science Conference a couple of weeks ago, I was listening to a talk where the speaker was espousing the requirement of a wet-dry cycle in order to get from the low organized carbon up to something that could be used by life. And the comment that he made was that wet-dry cycle was critical for the emergence of life, which is great for people who are fans of looking for life on Mars, because it helps direct your search a little bit. But it was a real Debbie Downer for those of us who like thinking about the little jellyfish in the oceans of Europa and Enceladus, because he flat out said, that those ocean environments are never going to have the wet-dry cycle that allows you to build up the organics that you need to eventually emerge as life. So I just wanted to know if anybody on the panel would comment on that.
Well, there's a difference between the environment in kind of the middle of an ocean versus the environment at an interface. And so on Europa, we have places at the ice-water interface, so at the very top of the ocean. And so as Brittany Schmidt has talked about, there could be water inclusions or sills or locations where there's smaller volumes of water within, say, an icy shell. Those are potentially places where you could concentrate stuff. But on Europa, at least, perhaps even more interesting is the bottom of the ocean, so at the ocean-rock interface. There, it seems to me that you could have all sorts of interesting concentrations and interactions. You could build up materials there, to higher concentrations, and then those could eventually be dispersed throughout the ocean. I definitely agree with you that if all you have is just a uniform volume of water with no way of breaking that up into smaller, concrete units somehow, that it seems like that would be very difficult to provide enough concentration.
Just to add to what you're saying, Cynthia, I think that what's extremely exciting about the Icy Moon, something I'm still trying to figure out, is the role of gravitational tides for those habitats, because these are going to be generating so much instability in this habitat, at least in some places, you have that. That is going to provide you with some different habitats and move things around. So what's the role of those gravitational tides in the biosphere on the icy moon? Also something that maybe we talk less about is the role of the changing phases of ices. That also releases energy. That changes things throughout the day, whatever a day is on those moons. So I think that there is plenty of room for having, you know, potential habitats and evolution of life on this icy moon. Just because of that, it's not an inert world. It's moving all the time, and it's pushed and pulled all the time.
You had to be careful, right? The water paradox for the origins of life arises because water is corrosive for certain kinds of bonds. So the NH2 carbon bond of cytosine is hydrolyzed in water to give uracil. It changes the information. You're living in an ocean of water inside your skin. You manage that by repairing, and the assumption is that in the origins of life, you could not repair because you had not sufficiently evolved Darwinianly to have repair mechanisms. So RNA molecule looks like a molecule that requires wet-dry cycles to have emerged. Now, suppose life did not have access to wet-dry cycles to emerge. Suppose it was perpetually underneath the water of an ice cap. Now, the structure of RNA would be different, right? The Darwinian biopolymer would have a different structure that would not be so easily corroded by water. Or you could go to Mike Russell's lab, right, and discover that, as you're saying, right, we have hydrothermal vents, we have all sorts of compartmentalization at the interface between the water and the rock. And they would have solved this problem in a different way, but in a way that would have led you to a first genetic biopolymer that had a structure different from RNA that you know and love. So the RNA that you know and love is giving you the signatures. It looks as if, I wish I knew how RNA were made, okay, prebiotically, I don't, but it looks as if it was made in the environment of a wet-dry cycle. But that's the consequence. That's the coevolution of the biosphere with the geosphere. And with the geosphere being different, sub-ocean of Europa, Enceladus, whatever, the biosphere will have different molecular structures that adapt to it.
Good, so I can continue to
You
look
can
forward to my little
continue.
jellyfish.
Absolutely.
Yes. You know,
Yeah, there's one additional point, and that is that you have to take into consideration the role of protective minerals. Not just the wet-dry, you know, drying and wetting and producing this buildup in sort of what I think of as a naked outcrop, but the fact that clays and many other minerals can actually physically and charge protect organic materials. Seems that you have a natural potential storage unit available for you know, building those up. So I think it's a weak overinterpretation of the chemistry to impute that to lack of being able to create life on ocean worlds. Let's see, Rocco, you had a question? You have the...
Actually had a comment and a question. My comment is back to Dale Andersen and Lake Untersee, where you said that it was, I want to make sure that people don't think that it's just a matter of isolation that has kept Lake Untersee and those lakes like that with a microbial ecosystem dominating. Because if you, it's energy drawn, it really is energy driven. If you were to put fish in there, for example, the whole ecosystem would collapse because there isn't enough energy to support that kind of system. And that happens regularly throughout different ecosystems here on Earth. Now the question I have gets back to one of the fundamental things that I've been trying to struggle with and think about for a long time, and that is with regard to Mars. Specifically with regard to nitrogen and the nitrogen cycle on Mars. Now we, and Steve, please comment on this if you would. I think that nitrogen is an important element for life. Mars has nitrogen, has a little bit of N2. But one of the things that has always puzzled me is why don't you find nitrates and ammonia on the surface of Mars?
We did.
We have worked out an abiotic nitrogen cycle that says theoretically you really should have, especially if Mars was warm or wet. Just wet. I don't care how warm it is, so long as it was wet. And if you don't have available kinds of nitrogen, such as nitrate and ammonia, or nitrite even, or amines or amides or something like that, you're going to find it very, very difficult to have evolved a living system. And to get the kind of chemistry you need, I think, to even start prebiotic chemistry. Now, I'm willing to listen to someone who says that you don't need nitrogen. It's not important at all. I have yet to be convinced.
No. You need I mean, if you look at a peptide backbone, you have carbon, carbon, nitrogen, carbon, carbon, nitrogen.
Exactly, my point.
Why we say we're carbon-based life form is just beyond me. And DNA, right, is carbon, 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 oxygen, phosphorus, oxygen, carbon, carbon, carbon. So the backbone atoms, right, it would be nothing.
Well, there is phosphorus there, but the nitrogen that's there on Mars is not very...
Let me get the facts straight here, because I don't think that...
Sorry, Rocco.
Go ahead.
Your prediction of nitrate on Mars was prescient. Congratulations. We did discover it, though.
But not very much.
100 parts per thousand. That's enough to grow petunias in.
Potatoes.
Why are there no petunias there?
Pardon? Why are there no petunias? That's a different question. They're not nitrate limited.
Seriously, there isn't sufficient, that was just a trace amount. And so, since I've been interested in this, I looked at it much more thoroughly and talked to other people, and there could be some confounding factors in that measurement.
Well, let me make the other question here. Right? You have ammonia on one hand, which is where nitrogen appears in very useful forms in building blocks, right? So in nucleotides, adenine, it's the ammonia level of oxidation. Say nitrate, nitrite. If I mix nitrate and ammonia, I have a disequilibrium, and I can eat it and get energy out of it. So the fact that we have perchlorate on the surface of Mars, right, is a statement that we have had an inventory of oxidation power. So I would expect to have relatively little ammonia. Of course, if there's a lot of ammonia and a lot of nitrogen in that state, that's a good biosignature for me because that's something we look for. But I suspect that we have a lot of nitrate compatible with perchlorate, but of course relatively little ammonia compatible with perchlorate.
And that's right. The nitrate discovered and characterized by salmon over almost a dozen sites correlates with perchlorate. And the concentration is high enough that things will grow in it. I think you could, 100 parts per thousand is not below the uptake limit of, unlike the N2 as you showed in experiments, the nitrate is not below the uptake limit of microorganisms at 100 parts per thousand.
100 parts per thousand, you mean 1% by weight is nitrate?
Not one.
100 parts per thousand or 100 parts?
Sorry, 100 parts per million.
Okay.
100 parts per million. Yeah, 100 parts per
We
million.
should rename it Planet Nitrogen.
Yeah. Sorry. It's just a factor of 1,000.
Still not very high.
No, it's not as high as a terraformer would like, but it's high enough for...
one would predict it ought to be.
Right, I agree with that, but it's high enough to not be a showstopper for life.
But a hindrance.
Okay, I grant you that. So, Rocco, why don't you keep the blue box on this side of the room, and we'll go to Ralph next while you find the next person on this side of the room that wants to ask a question.
Ralph.
So my question for the panel has to do with the second genesis in the outer solar system. It seems to me the distinguishing feature there is that you've got ice-covered oceans. The Earth may not have been a snowball, so if there's a difference in the genetics or whatever, it would seem to have most thing to do with the physics of forming ice, life with ice. And I mean, everybody in the panel seems to think you're going to have hydrothermal vents in the bottom of the ocean, and we have that in ours, so that can't be the difference either. So my question is there something about the physics of prebiotic chemistry and ice that's going to be telling, or do we know anything about that?
As someone who's not a chemist, I guess I would say that, yes, it's true that I think that's a really good point, that perhaps if you have a second genesis, a second origin of life on an ice-covered world like Europa, that the distinguishing factor could be that life formed under, that life formed under an ice layer. But I think that more likely the life is not going to be forming in the ice layer itself. So probably you're going to have interactions with that silicate layer, that rocky layer at the bottom of the ocean, that create, maybe either there are those hydrothermal system analogs, but maybe those materials are brought up again to the top of the ocean, so to the water-ice interface at the top of the ocean, and then you also get materials that are created at the surface through radiation processing, which could work their way down. So that layer there at the ice-water interface could also be an interesting location for concentrations of chemical products, perhaps for some interesting chemistry to take place. I don't think you can necessarily say much about whether that would make the life completely fundamentally different from how life formed on Earth, or whether it would have similarities. But I think that that's an interesting point.
Yeah, I mean, one of the foremost important words in science is "I do not know." But you raise an interesting point. The question is, how do we know that the Earth was not a snowball, right? And it's quite, there's a whole line of argument that says the Earth was a water world, that is, there was no dry land at all. And then there's also this notion that the solar activity was a quarter of what it is today, and therefore the Earth was frozen. The problem that and so it may very well be that the origin of life on Earth is exactly the origin of life on is that your point?
Well, that's part of it too.
Yeah.
So it might be exactly the same origins problems. Now, the only thing wrong with that is that Dustin Trail and Steve Moises, and there's people who look at these very old zircons, have told me that there is liquid water, ocean exposed, maybe landforms, dry area, roughly twice the area of the state of California. So there is some experimental evidence, which from old rocks that were there, that have survived, that would argue that maybe this is not the same thing. But there are certainly ways of analyzing these global picture and Earth accretion history to say that yes, Earth was just as much a water world frozen with an ice layer as Europa is today.
Ralph, why don't you pass your blue box back that way, and we'll go to this side with Bill.
Yeah, Bill?
So I have one observation, quick, and that is, unlike Carolyn, I'm totally able to grasp the concept of the box. No, that's okay. And then second observation is, I don't know what Chris had for lunch, but I'm having that tomorrow. He's totally on his game. But I thought might just be fun as an exercise to go down the panel, since we're here talking about candidates like Enceladus, Europa, Titan, Venus, Mars, et cetera, and just get the opinion from everybody except Cynthia, who's already weighed in. Where do you think it's most likely? What do you think is the best candidate?
What's your first choice?
What's your first choice?
Where would you go first if you had a little spaceship? So why don't we start with Carol?
Ranked order voting.
What does that mean? Okay,
So,
Carol.
I think I said it to begin with. I think the lowest hanging fruit, the easiest place to find life, evidence of life beyond Earth, is Mars. And that for several reasons. One is that we have a program that's going there on a regular basis. One is that we plan to send humans. It's always the next thing that we're going to do, but and before we do that, we really kind of owe it to both life and human life to find out if there's life on Mars. And so maybe it does share a common ancestry. So what? Yes, maybe it doesn't, completely alter our view of the origin of life, but maybe it does. So I would say it's Mars.
Cynthia, we already know your answer.
I already said Europa, but I'd like to make one other point, which is that when I used to run a student astrobiology summer internship program at the SETI Institute, I would always give the students kind of an astrobiology of the solar system overview talk at the very beginning. And I would go through, all the places in the solar system where there might be life. And the place that they always found the most compelling and the most surprising was actually Venus. And I would go through a lot of the evidence for possible habitability of, really interesting bits about cloud layers on Venus. And that was the one that none of them had ever thought about before. So, Europa's my favorite place, not to diss Mars, but I think we should really take another look at Venus.
You changed your vote to Venus.
No, I didn't.
So you went for Europa?
Europa's my favorite place. Mars is boring and old and done.
I'll let you to deal with that.
Boring, it's too easy.
I think we have to take a step backward here and think about, what's really driving the answer. If the answer is that Mars is the closest planet to ours, the one that we have an integrated program, and we have put the most effort and we know the best, then maybe Mars is the best place to go because we have all of that information. Now, if you think how, what is the world where I have the possibility of getting the answer faster, maybe it's going to come as a shocker for everybody, but I think Enceladus is our best choice.
Doesn't shock me.
It just depends what you want. If you want the best shot at getting something that's coming from very deep, right there without too much effort, it's Enceladus or some geyser on Europa, if we have geysers on Europa. On Mars, we already have the map of the biosphere in our heads. And frankly, I'm going to shock more people yet, but I don't think that the landing site we have in mind might be the best place to go for that, because the engine of the biosphere on Mars always has been at the equatorial region, and then maybe this is not for nothing that we have those methane emissions right there. But if you want to go to Mars, you will have to put a lot of effort to get into that biosphere, which is a big drill, which will require maybe humans to go there, et cetera, et cetera. So it depends very much what you have in mind. For a quick grab, best shot, Enceladus or Europa. For a long-term endeavor, Mars, definitely, because we have the best background.
Okay, so we're putting you in the Enceladus category. Well, that was your, if everyone gets one word, it's Mars, Europa, Enceladus. Steve.
Well, Chris, you're not going to like this, but I will make a prediction that almost everybody here in their lifetime will see a life form which does not share common ancestry with the people on the stage, and it will emerge from a chemistry laboratory which is creating it.
Okay, Mars, Europa, Enceladus, lab.
His lab.
We might be, but it might very well be in Paris. I can think of a number of places where it might be.
Yeah, that's a good answer. It's a good answer, Steve. You're right, I don't like it, but it's a good answer. Bill?
I think it's sad we have to choose.
Okay, but you were told that it was a framework of your boss.
I think that Mars would be a great place to go, but I think it will take, like on Earth, in the search for early life on our own planet, it'll take humans to find evidence of life on Mars eventually. But right now, Enceladus is offering it up in the plume. It's just waiting for us to come get it.
We have 2 votes for Enceladus. Penny.
So I am a modern superwoman, or so I like to think, and I want it all. So I am looking forward to the session on non-traditional propulsion, because really, what we need is a cheaper bus ticket so we don't have to make this decision. I want us to be able to get to the outer solar system, for less than billions of dollars. I want us to be able to have repeated missions to outer solar system objects, not just one. So I guess my favorite planet is planet advanced propulsion.
Yeah, I guess the other part of that equation, too, is it'd be nice to get data back before we're all gone.
I don't plan to die until we get the data
okay.
back.
I'll take the last vote. I'll vote for Titan. If I had a spaceship, I would go to Titan.
It's pretty
compelling. The argument there, I don't care, I'll go there anyway. The argument, my rationale is, I'm betting it all on the extreme weirdness of life on Titan. I'd rather risk it and go for the weird than play it safe and go for what Alfonso calls the close cousins. So the weirder the better for me. That's just the way I am.
You should admit that you just want a wingsuit.
The story of your life.
Yeah, exactly. Exactly. So other good questions. Let's go to this side of the room. Yeah.
And then there's one back here.
We got 10 more minutes.
It is now. So a call from Planet Advanced Propulsion, but I'm going to ask this in a deliberately provocative way. If all you want to do with the solar system is look at it, the rest of us would like to borrow it for a while. There are things to do with these bodies other than science. And a significant limiting factor that's inhibiting those of us in the commercial world from doing some of those things is people like you don't like it if we think about landing, because you're afraid that we're going to contaminate the purity of the science, although we manage to do biology on Earth with biologists. Is there any point at all, short of the entire GDP of the Earth is spent for the rest of time studying that planet, at which we can say enough is enough, If there's life there, it's hiding, and we're not going to find it without sending people anyway, so it's okay to go now.
Is the moon enough?
Good question.
Okay, you can have the moon, you can take the rest of the solar system.
There's a lot of asteroids. You can have some asteroids too, as many as you want. You know, just leave us one of each.
A quick follow-up is, you know, if your answer is, we'll just keep studying it forever because we can't rule out that we know, realize that in spite of what you all may think, the solar system is actually not a scientific preserve by any kind of international treaty. You know, if a reasonable threshold can be set, there's a chance that it will be observed. If a reasonable threshold is not set, it probably will not be observed.
But maybe you can clarify something for me.
Paralyze your stuff.
Let me frame that question, because I think you can divide the solar system in context of your question in 3 categories. The moon on one end, the outer solar system on the other, which is really far for exploitation or human exploration, and Mars, which is really the key point of contention. And so if we focus your question on Mars, it's a very interesting question, especially in light of recent announcements by people like Elon Musk. They're going to send people to Mars, right? In fact, he already sent his car. It'll be waiting for him when he gets there, right? And so Carol, who's been, as she said, very interested in human exploration and also interested in the search for life, is probably in the best position to lead off the answer to that question, focusing on Mars. We're not talking the whole solar system. What's at play here is Mars. How long do we scientists try to keep the contaminators off of our planet?
So I think that we really are in a situation where the more you know, the better you can make a investigation that is not going to be just a shot in the dark. I think we made a bad mistake, not that we didn't learn anything, but by going straight to a life detection mission on Mars in 1976, we sort of killed the Mars program for 30 years because, you know, people interpreted the answer as we didn't find life on Mars, and so that means there isn't life on Mars, and, you know, let's go look somewhere else. But in fact, we made it in a very ill-informed way. So now I think we have a much better understanding of what is the surface of Mars like, what is the subsurface like, where are the good environments to go look, and thanks to the Ocean World Program, we're actually starting to build instruments again that are capable of doing life detection. Because that's the other place that we killed for 30 years, or 40 years, is we never built any life detection instrumentation. So there's nothing to go look with. So I think that we now are in a position where we can go to a few well-chosen places and we can look. If it's not there, then we have a pretty good confidence that we really don't have life on Mars, there is no conflict. If it is there, then we need to understand it better. So, you know, we need to understand its biochemistry. We need to understand whether it is, in fact, shares a common ancestor with life on Earth, whether it can interact with life on Earth, whether if we bring it back to Earth, it's going to disturb our own biosphere in some way. So I think yes, it makes sense to look. And I think there is a finite number of investigations. I don't want to hazard a guess as to how many, a minimum of 1, that we can answer the question.
Okay, speaking of questions, we have time for one last question. Sorry, so the guy here holding the blue box, go.
I'm Ramona Samaskis from Beagle Lab for Ocean Sciences. I was fascinated by the Venus talk this morning and the prospect of life in clouds. That these gas giants in our solar system with many more deeper clouds, but there's no talk about searching for life there. Is there a good reason for that?
Carl Sagan had the big, gas, the floaters in Jupiter, right?
Sagan and saltpeter in 1976.
Let me suggest you ask David Grinspoon. Yeah, why don't you answer that question, David?
Cloud-based life is, something that I don't think enough attention has been given to, and indeed, it has been proposed there could be life. As you mentioned, Sagan and saltpeter had this idea about floaters and sinkers and this whole imaginary ecology and Jovian planets. But one advantage Venus has potentially is interaction with the surface. It seems as though the surface is volcanically active, and in fact there are active geochemical cycles. There's a sulfur cycle involving volcanism and transformations happening in the atmosphere with solar energy, and so on Earth certainly life is something that rides biogeochemical cycles. And on Venus you have that potential, and I'm not sure you have that potential on Jupiter, but you certainly have other sources of disequilibrium such as lightning and so forth, so it's worth thinking about.
Great, okay. Let's thank the panel.
Yay, panel.
And,
thank you, panel. Thank you.
Just